so of it. If you look into the definition about the plant tissue culture is, tissue culture is the term used for the process of growing in the laboratory. It means to tell that you can grow the culture in the laboratory, that is in vitro or in the glass vase. This is the meaning of artificially in. So you can also have to note that if I have to grow a cell or a tissue in the laboratory, develop it into a fully grown plant, I should condition for it. That is all the environment which is efficient, available for it, the nutrients required for the full development. These tissue cultures, whenever we are doing, it is going to produce clones of the same of the plant, in which all the product cells have the same genotype. So what it means to tell is, we know that clones are all those plants which are genetically identical. Okay, they are produced asexually. In other words, when it comes to the plant, we do what? Vegetative propagation. So this is the meaning of the clones. That is, they are genetically identical. They are produced asexually. So when we look into the history of plant tissue culture, the cell theory was discovered in the year around 1838 to 1858. So during this period, two German scientists, Schiltin and Schwann, they discovered or laid the foundation for the cell theory, which is universally accepted, that living organisms are made up of cells. So later on, this laid again to the foundation for the totipotency. What do you mean by totipotency? The ability of an individual cell to develop into a whole plant is referred to as cellular totipotency. In other words, whatever the cells or the tissue which I am going to grow in the laboratory, it is going to develop into a whole plant. So this phenomena of that cells and tissues under the appropriate condition, what we are providing, and then it develops into a and fully grown plant, is known as the totipotency. So when we look into the history of in 1902, another German scientist named as Haberland, he is known as the father of tissue culture. He did lot of discoveries in plant tissue culture. And what in his experiment, what did he do was, he took fully differentiated cells under artificial media. When he observed that, he found that the cells were getting enlarged. That is, they were undergoing thickening, increase in size, but never divided. And this was a failure in the experiment, because there was no meaning to totipotency here, because the cells did not divide to develop into a fully grown plant. So ultimately his experiment failed. But nevertheless, he speculated that there is something missing in his media, and he called it as growth enzymes. The growth enzymes are missing in his media, and that is why the cells were not able to divide. Today we all know that growth enzyme, what he mentioned, is the growth hormones. So look at the intelligence of this guy, that how he assumed that what could have gone wrong, and that was in the year 1902. So another failure of the experiment was, he took highly differentiated cells for the growth of the cells. And then whatever the media what he used were very simple media, that is the salts and the sugars, the sucrose what he used was very negligible amount of nutrient was present, which was not sufficient for the growth of that or division of that cells whichever he took. Then way back in 1904, Hanning, he chose embryonic tissues to culture. So what did he do? He excised or he cut the mature embryos from the seeds of several species of Crucifera plants, and he successfully grew them to maturity, containing a media which consisted of mineral salts and sugar solution. And this was a successful experiment. And in 1908, Simon regenerated callus, buds and roots and established the basis for callus culture. Now we shall deal in detail what is the meaning of callus, define what is callus in our future classes. But for time being, just remember callus are undifferentiated mass of tissue. Okay? They are a parenchymatous cells. When we provide proper nutrition condition, the environmental condition to this callus, it can in turn develop into a fully grown plant. That is what is the callus mean. So Simon was able to produce the callus and regenerate the buds and roots established for the basis of the callus culture. In 1922, two scientists independently pursued their experiment on the root tips. So we know that through the roots and whole plant can be generated. And this is what these two gentlemen did. Robbins and Cote independently pursued their experiments and reported the growth of isolated root tips. So going a step further, in 1926, W. White, F.W. Wendt demonstrated that there were growth substances present in the plant. That is, he took an example of the Avena plant and he studied that there are what are called as growth regulators or growth promoters or growth hormones. And he was able to isolate auxin. Just remember this word, auxin. In 1933, other scientists, they were able to isolate indole acetic acid. This indole acetic acid is a form of auxin. In 1934, there was a big breakthrough in the plant tissue culture. A scientist named White generated continuously growing culture of meristematic cells. We know that meristematic cells are actively dividing cells. Okay, so growing cultures of meristematic cells of tomato on media containing salt, yeast extract, and sucrose. And then later on, the same scientist, he came to a conclusion telling that instead of using yeast extract, okay, we can use vitamins. And what were those vitamins? It was the pyridoxine, thymine, and nicotinic acid. So instead of yeast extract, an alternative vitamins can be used. And so that he established the importance of additives. Additives means are these substances what we mentioned. So into your media, you're adding these substances. So these substances are known as additives. And also he developed the media for the root culture. And today it is known by his name as White's media. Whatever the root culture media is there, it is known by his name. Then coming to 1939. Now, like, just look at the years, girls. Like, simultaneously, you can see that parallelly in most of the country, the experiments was running about, okay, going through. So it was almost a quick discovery. If one person started, you can see how fast the experiments were being carried out. One was doing about the callus culture, some the root culture, some the embryon culture. So it was going through a fast pace. So in 1939, Gotteter successfully was was able to continuously grow cambium culture from the carrot and tobacco, and he showed that it can be maintained for several months. Today, how easily we can grow this carrot and tobacco, but look at in 1939, they were able to take the cells from the carrot and tobacco and then develop it into a whole plant. And also they told that this indole acetic acid, which we spoke previously, what is indole acetic acid? It is a form of an auxin. What are auxin? They are the growth promoters. So he gave a conclusion telling that this indole acetic acid promotes the root growth. Okay, so the two important parts in the plant is the shoot and the root. So he told that this IAA promotes the or propagates the root growth. Similarly, the carrot explants develop into undifferentiated mass, and it can be maintained by repeated subculture. That is, he told that he took the explant. What is an explant? Explant is a part or a piece of a plant. So it can be your leaf or your embryo, your cambium or the root or the flora, the bud. Any part of that plant is known as the explant. So he took that explant, and from that he was able to develop the callus. What are callus? We defined it as telling it as an undifferentiated mass of tissue. So another step ahead he went. He told that we can maintain this callus, okay, for many months. We can keep it alive. How? By a process known as subculture. Remember the term subculture, because in a future class we'll be explaining in detail about the subculture. So I can maintain my callus in the media for many months by repeatedly subculturing. Then in the year 1943 to 1950, we see that there was discovery in the tumor inducing principles of crown gall tumors were identified. That is to say that, so we all know what our tumor is. Okay, that is the cells divide repeatedly, uncontrollable division. So the same was observed, this uncontrolled division was observed even in the plant. Okay, so this was known as the crown gall tumors. Later on, they identified this tumor in this plant is caused by an bacterium known as agrobacterium. And this agrobacterium was a groundbreaking step in the agricultural field, because today, like, we are using all this for the experiment in whatever we are doing in the plant tissue culture. We are using this agrobacterium, and in detail again, once again, we shall be studying about the agrobacterium. Just for now, you remember that agrobacterium is a bacteria which infects the plant. It is present in the soil, and then it infects the plant, and it produces the crown gall disease in the plant. That is a continuous division of the cells. In 1948, formation of adventitious shoots and roots in tobacco was predicted by scientists Shug and Swine. Throughout the dates you observe, okay, these are the periods when World War was going on. And but on the other hand, we should appreciate the anticipation and the speculation of our scientists, what interest they showed in the field of science, not only in plant tissue culture, but in all field of science were glooming. Okay. So again, independently, you see that Shug in 1944 and Swine in 1951, they reported that tobacco in the pig tissue culture, the addition of adenine and high level of phosphate favor the callus growth and bud formation even in the presence of indole acetic acid. What it means to tell is they independently found out that they were able to culture the pith tissue from the tobacco plant. How did they culture? They added what? Adenine. Adenine is what? It is a nucleotide. Okay. So it is a base, nitrogenous base. They increase the concentration of the phosphate. So increasing the concentration of the phosphate, even in the presence of indole acetic acid, they saw that there was a formation of the callus growth. What did we tell about indole acetic acid in a previous section? We told that it was proved that indole acetic acid is responsible for the root formation. Now, what if these scientists are telling that, okay, if indole acetic acid is present also, it favored the growth of the callus. Okay, that is, they gave a picture telling that though there is indole acetic acid, if I just increasing the concentration of the phosphate, it helped the callus formation. Okay, and there was no that development of the root. So you can just manipulate your media, you can manipulate the ingredients in your media, you can play with your media. Okay, you increase one substance, decrease another substance, it's all trial and error, and then they observe that what it favored. So in 1954, Jansky and Scoop observed that the division of cells occurred only if the cambium tissue was present. Pith cell alone did not show any division. It means to tell that some of the cells cannot divide independently by themselves. Okay. For example, pith cells, they did not divide by themselves independently. But in turn, whenever cambium tissue was present along with the pith cell, they showed the division. That is the meaning of this sentence. Then F.C. Stewart in 1948 observed vigorous proliferation of carrot explant due to the presence of the stimulating substance in coconut milk, which was not an auxin. So what I meant to tell was that they used coconut milk as a media, one of the ingredient in the media, and they came to a conclusion telling that this coconut milk was not an auxin, but still it stimulated the growth. So then what happened was later on the scientists, they also started using the coconut milk in their experiment. So because this also was able to give a continuous growth. He also discovered the somatic embryogenesis from carrot cells in suspension after being plated on solid media. That is like somatic embryogenesis. What we mean, we already know what are somatic cells and germ cells. Okay, so he took the somatic cells. Okay, from where he took it? From the carrot. Okay, he took from the somatic cells from the carrot, and he was able to develop the embryo, the development of the embryo. And later in 1955, Miller isolated the first cytokinin and kinetin. This also is also a important growth hormone. So you can see that auxin was identified or isolated, and also we see that cytokinin, kinetin, indole acetic acid, these all were isolated and identified. These are what growth promoters or growth hormones which are naturally present in all the plants. Please remember that. Okay, in any plant you will find these all. So what does this tells us? That is, if it is present in our natural plant, when you're growing the plant or when you're growing the cells and the tissues in the laboratory, you have to provide these growth hormones. Only then the cell division can happen, the cell proliferation can happen. Okay, it may be a cell, it may be a tissue, or it can be an organ, it can be a callus, whatever you're taking. Okay, you provide the media, you provide the growth hormones, and you provide the proper environmental condition, and that can lead to a development of a whole plant. And this word totipotency, this word was coined by the scientist Stewart. Okay? Please remember that also. In 1952, the virus-free Dahlia through meristem culture was developed. This is another important aspect. You can see that you are developing, okay, a plant which is free of virus. How? Through using the meristem culture. So keep this point also imprinted in your mind, because this also will come across in further how we are going to produce a virus-free plant using a meristem culture. This was followed by the micrograft, and then in 1957, once again, Zug and Miller, they discovered that the root or the shoot formation in the culture depends on the auxin is to cytokinin ratio. Okay, we told, now you are familiar with these two words, auxin and cytokinin. That is, if I increase or decrease the ratio between the auxin and cytokinin, okay, either the root or the shoot can be formed. In other words, okay, auxin enhances the root formation, okay, and the cytokinin enhances the shoot formation. This is what they meant to tell us. You just play with the auxin and cytokinin ratio, and either you can induce the root or you can induce the shoot. Then in 1958, we see that the embryo, pro-embryo formation in callus, clumps, and cell suspension. 1960, enzymatic degradation of cell wall for protoplast formation by cocking. That is, he used enzymes to degrade the cell wall. Now, what happens? We know that the plant cells are made up of the cell walls. If I degrade this cell wall, what is left? Protoplast is left. So, enzymatic, how I'm going to degrade the cell wall? By using the enzymatic degradation. And this was again one of another breakthrough field in the scientific field, this enzymatic degradation for the protoplast culture. Then in 1916, there was a vegetative propagation of orchid by meristem culture. And in 1962, we see the development of MS media by Murashik and Shuk. The word MS stands for the letter Murashik M and Shuk S. Okay, that is what is known as the MS media. So this MS media is a potent nutrient which is still now used in all plant tissue culture laboratories. Okay, it is a hallmark media for growing the plant tissue culture. I'll be saying the importance of these media, and few more scientists we will shall speak about in our next journey about the history of tissue culture.